what I want to talk about is the work that we've been trying to understand mechanisms that contribute to the formation and processing of memory, biological mechanisms. And I'm going to do that by discussing a particular structure, the hippocampus. And as we go through this, I think you'll see why we focused on this. And there's just a couple of sort of historical notes. This is a cutaway view of the rat brain showing the hippocampus. Again, I have to remember that I'm on Zoom here, so I can't point to the screen. I'm going to use the pointer. And so you can see overlying neocortex removed to reveal the hippocampus. The hippocampus lies in the medial temporal lobe. It's sort of this, well, it was referred to as a hippocampus seahorse-shaped structure, but it's sort of this bean-shaped structure that is right in the center of the brain. As you if you curl your fingers, put it to the side of your brain, the temporal lobes, and you push it in, that's where the hippocampus resides anatomically. Functionally, the hippocampus lies at the top of a processing hierarchy. Information comes into the brain, it goes into primary sensory areas, it goes to higher order sensory areas, polymodal association areas, where you're kind of integrating information. You can sort of see there's a kind of a flow of information into the hippocampus via the temporal lobe, so it kind of comes in, comes around into the structure that feeds into the hippocampus, the entorhinal cortex, from the entorhinal cortex to the hippocampus, and then back out again. So if you were to put it in like a pyramid, what you would see is that information from across the brain converges really at two primary nodes. One, the hippocampus, and the other would be the prefrontal cortex. You sort of think of the prefrontal cortex, it's the CEO, it's the executive, it has oversight over everything, and uses information across the brain to direct behavior. The hippocampus sits at about the same point in the hierarchy. But the question really is, the hippocampus is not acting as a CEO. The hippocampus is not directing behavior, but the hippocampus is directing the processing of the information. Maybe it's like what's the chief information officer? I don't know, whatever the chief technology officer. It's the executive that is responsible for the processing of information so that it can be used to direct future behavior. And so the question is, how do you process information derived from experience in the world to maximize its use in directing future decision making? That's really just that's sort of the core question. And sort of Boris introduced the topic. He said, I study memory. I always like to describe my work as not really studying memory. It's the study of understanding what memory is, rather than studying the mechanisms of memory. It's really to try to understand what is memory. We sort of think of memory as it's a simple process, right? Like in your computer, storage and retrieval, right? You get information, you store information. Later, when you need it, you retrieve it and you use it. Well, that clearly is not the way biology is actually conceived of memory processing. And there are a couple of reasons for that. One, this is sort of the big data problem in general, machine learning, AI, and computation, kind of artificial computation. And that is that you can solve a problem if the answers are buried in vast amounts of data. And your objective is to sift through that data, find the answers, and exploit that. In a biological context, well, the problem is, how do I actually solve the problem of the world? And your sampling of that problem is not dense, it's sparse. You get very little data. You may have no explicit example of the problem that you're going to solve. For instance, there's a predator, you're in an open field, what do I do? The big data solution would be send lots of organisms out, have them explore the space, see which ones get picked up by the predators, feed that into a large deep layer network, and it will solve the problem. What should you do when you have predators in an open field? If you're an organism, you don't have the luxury of that. And so you have sparse sampling, and you have to make judgments, predictions about expected outcomes based on prior experience that may only be generally related to the future applications. And you can think of the future applications as being described as a context. The context is the set of or configuration of features which really define the problem and direct the potential solution. So what you have to figure out is, how is one context, the features that you've actually experienced, how do they apply to a future context, a new problem. And that, ultimately, I would argue, that is the problem the hippocampus is trying to solve. How to select from the configuration of features in a space that you are experiencing, how to transform that into a representation that can be generalized into new context, which you can then use to predict outcomes, and then ultimately feed that up to the executive, the prefrontal cortex, for example, so that it can actually direct behavior to solve the problem. The hippocampus figures out what the right information is to solve problems that will be encountered in novel contexts. So I sort of summarize that as it's the expression of adaptive behavior in novel contexts. That's the problem the hippocampus is trying to solve. How do you do the right thing when you don't actually know the answer? You don't have the ability to look it up in your previous database because that database is too sparse. So how does the hippocampus do that? And ultimately, what's the structure and the nature of that data the hippocampus is collecting? So there's been a lot of interest and focus on the hippocampus as kind of a memory storage and retrieval device. You sort of think of it as identifying features, taking those features, storing them, perhaps consolidating them, transforming them into another form that's more efficient, and then retrieving them. Again, the storage and retrieval of static information. You can think of this as the general recognition problem. And that is, how do you recognize when you've already seen something? How do you retrieve that, and how do you use that? But there's another aspect to the use of information, which goes beyond storage and retrieval, and that is the use of information to develop simple predictive models. So ultimately, again, the storage and retrieval model, it's limited largely because it relies on adequate sampling, but it's also limited in that it requires that the previous sampling actually correspond in some way and matches the present context. So if you don't have that kind of exact match, if you can't look up information, then you have to develop a model that's capable of doing prediction based on that information. So if the idea is that you want to build a model rather than simply store and retrieve and do pattern matching, what is that model, what's that predictive model going to look like? Well, first of all, it's going to have to identify the relevant features, and then it's going to have to determine how those features might be causally related to some future outcome. And that is, it has to establish the kind of causal dependency, which can only come by both kind of storing, retrieving, and evaluating temporal contingency, or keeping track of time sequence information. It's not whether A occurred, it's not whether B occurred, it's did A occur before B, and is A now predictive of B, and can we now develop a model that takes features of A and then uses them to predict outcomes in B. That's the objective. So first step is you have to encode, and you have to be able to retrieve time sequence information. And just thinking historically, again, the hippocampus and its involvement in memory formation, there are two things that kind of stand out. One was the seminal case of the patient HM who underwent in the 1950s a surgical procedure involving bilateral resection of medial temporal lobes, took out his hippocampus. HM was unable to form any new memories of experience, life experience, things that you have done. So that suggested that the hippocampus was critically involved in a form of memory, episodic memory. It was really about memory relative to the self. But a part of that kind of event memory, episodic memory, was the notion that events actually have this kind of time sequence structure. And you kind of fast forward from the 1950s, where there's a lot of work on hippocampus and this kind of episodic memory, to the animal work. And John O'Keefe in the early 1970s, doing recordings in the hippocampus of freely behaving rodents, rats, discovered that individual cells in the hippocampus respond to different locations in space, so-called place cells in the regions of space he described as place fields. And damage to the hippocampus impaired another form of memory, spatial memory, impaired spatial navigation. So the features of those two forms of memory, episodic memory and spatial navigational memory, you can think of as this, the kind of temporal sequence dependency. In navigation, it's not enough to know where you are. It's not enough even to know, to have a map of where you might be. You have to be able to take that spatial information, sequence it appropriately so you can plot a trajectory from where you are to where you want to get to. Again, solve a problem involving the evaluation of temporal sequences. So thinking about temporal sequences, temporal sequence memory, what would be the best way to study that kind of memory processing? And the approach that we've used is the application of what's shown here, and that is multiple single unit extracellular recording. In short, you take a wire electrode, you drop it into the brain. So this is a kind of a tiny electrode, 35 microns in diameter per scale. You pull one of your hairs, typical hair, thick hair might be 100 microns, fine hair might be on the order of 30 microns. So this is about on the scale of a very fine piece of hair. You drop it in. The wires are insulated except at the tip. The tips are exposed. The conductors can pick up the electric fields generated by nearby brain cells. And among those fields, some of them will be generated by synaptic currents coming into and going out of dendritic compartments, but some of them will be currents that reflect the generation of, as shown here, action potentials. So spiking activity, which typically involves very small or short dipoles, and that is that it's really hard having to point on the screen, sorry. But action potentials, which typically are generated in particular regions of the cell, the initial segment of the axon, or in the vicinity of the cell body. You can think of the cell body as being the source of action potentials, that the currents involved in that form very short dipoles, that in order to see short dipoles, you have to be close to those current sources. And so what that means is an electrode dropped into a medium containing many of these cells is going to pick up the synaptic currents of a large population, what's referred to as the local field potential. If you were to put the electrode very far from these cells, you might refer to it as the EEG or electroencephalogram, but when you place it here in the vicinity of cells, you're going to pick up the macroscopic local field potentials, but then the action potentials generated only by cells in the nearby vicinity in a sort of a radius of maybe 50 microns. And a typical cell diameter, 10 to 20 microns. This is approximately the scale. This is approximately the scale over which you might actually see it. So it's just, well, not that whole, sorry, my mouse movement is limited. Just cells that are near the tip. And so what that means is if you drop in a number of these, and of course the packing here, I'm showing you, this is a cartoon, the actual packing is much denser. So you're gonna have a large number of nearby cells that are going to be generating action potentials, which you will pick up at the tips of this electrode. And this electrode has, as you can see, 4 independent conductors, 4 wires. And each of those conductors is going to pick up these action potential electrical fields. And the field that it picks up is going to have an amplitude that is dependent on the distance. The farther away it is, the smaller the field. And so you can use this approach, 4 wires, where you have a distance-dependent amplitude to nearby sources to identify and resolve the contributions of individual cells. Basically, you can separate multiple sources, different cells, separate them, identify them. If you put in a bunch of these, it means you can record a bunch of individual cells and the action potentials that are generated by them. That's the idea. And this is that was a cartoon. This is the actual, this is a sort of a CAD view of the device that allows the insertion of multiple multi-wire electrodes that are shown on the left, which we describe as tetrodes, 4-channel electrodes. And this is the kind of data, that you get. This is actual data. This will be collected over maybe 10 minutes. Each of the points that you're seeing here, each of those little dots is a single action potential event. Each event looks like what you see up here on the left, and that is it's about a 1 millisecond window. That's the voltage trace recorded at each of these 4 recording sites. The voltage trace over about a millisecond, a little over a millisecond. And so you pick up an action potential event, 4 locations. And what you can see here is the amplitude of that event. It's the same action potential, but it has a different amplitude on the different channels. And that's because each channel is a different distance from the actual source of that. And so if you plot here the amplitude of each event on each of the 4 channels, I'm just showing you 2 channels here, but there are actually 4 channels. Each one you'd have 4 dimensions. What you can see is that these clusters form. So here, for instance, the green, the green events cluster because each of the action potentials within that cluster have a profile in which they have a large amplitude on channel 1 and a smaller amplitude on channel 2. What that means is the cell that's generating these events is close to channel 1 because it's large amplitude and far away from channel 2. And so you do this on all 4 channels, you can essentially triangulate the effective location of all of these different sources. Different clusters therefore represent different source locations in 3-dimensional space. And the theory behind it is that in principle, 4 channels, 4 non-coplanar channels gives you unambiguous resolution of any point in 3-dimensional space. That's the idea. If I have just 1 channel, and a lot of conventional electrophysiology, you put in 1 channel, you just record everything. Well, the problem is, if you think about 1 channel, that at 1 channel, any cell or action potential that is at the same distance to that 1 point is going to look exactly the same and have exactly the same amplitude, and you will not be able to resolve that. And so if you just imagine an imaginary string of a fixed distance, you swing that string around, and that traces out a sphere. That means any cell on that sphere cannot be resolved. It's going to be confounded. You're going to mistake, all of those cells for 1 cell. If you had 2 channels, now I have 2 strings, that traces out a circle. So any point on that circle, any cells on that circle, again, cannot be resolved. You add 3 points, so 3 strings. Now those are constrained. There's like 1 point and its reflection on the other side. So you go from a sphere to a circle to 2 points, and then you add the 4th, that gives you the 1 point. So that's the principle. So why have spikes? So currently there are a lot of methods for kind of extracting large-scale brain activity data, about activity in the brain. Some of those will include indirect measures. For instance, fMRI, that's using blood flow measures to sort of assay distributions of energy consumption, and that is you sort of infer areas that are metabolically active must also be neuronally active. There are imaging approaches, and that is, for instance, looking not necessarily at the action potential events themselves, but for instance, the ionic currents that might be associated with that. For instance, calcium imaging, looking at calcium flux as an indirect indicator of individual direct measurements of neuronal activity, simply not direct measurements of action potential activity, action potentials that reflect the communication or the output of the neurons. So why not just use those? They have the advantage that you can use, for instance, you can put in little mini scopes or little cameras. You record the activity not of tens or hundreds of neurons, but potentially thousands, tens of thousands of neurons. That would seem to be better. Well, it may be better. I mean, you gain the advantage of scale, but you lose one important feature, and that is the temporal resolution of the action potential events themselves. Here, you're not seeing spikes. So somebody's got to convince you if they're saying, I could get 10 times as much data. I don't have to make these little wires. I don't have to build a complicated device. I just put a little camera on their head. Or maybe I don't even put a camera on there. I just put like an EEG device or, put the animal on a magnet, do some functional imaging. I don't need spikes. So Wilson, you got to convince me that spikes matter. So that will be my objective, to convince you that spikes matter. And the thing that spikes have, because they are very brief in duration, they have the potential to communicate timing information. And that is, the time of occurrence of a spike could be used to encode timing information. And the sequence of spiking could therefore be used to communicate information about temporally sequenced events. And so what I will show you is that in the hippocampus, that's precisely what occurs. Not only do you see precise timing information preserved, but there are biophysical mechanisms that can take kind of continuous behavioral experience and transform it into a temporally sequenced format for subsequent use using the intrinsic biophysics of the system. In particular, exploit the properties of dynamic rhythms in the brain. And so it will sort of focus on mechanisms of encoding at the level of individual spikes, but then the leveraging of brain oscillations. Brain oscillations are a topic that have come up in the study of the brain because you see them everywhere. There are particular frequencies that seem to correspond to particular behavioral states or particular cognitive functions. And one of the capacities that oscillations have is that they have sort of a localization, and that is that, as I pointed out to you, the oscillations that record tend to reflect the activity of synaptic currents, and that is, they tend to reflect the timing or organization of inputs. And so if you think about why would you want to organize the timing of inputs, well, if you have a continuous system, in other words, you don't have a system like a digital system where you have it clocked, you say, okay, time step 1, we're going to collect information. Time step 2, we're going to perform some computation on it. Time step 3, we're going to transmit that information. You have a continuous system, and so in a continuous system, you need to have some kind of clocking mechanism in which all the elements that are contributing to that computation are coordinated. So there's this idea of coordinating computation within a system, but then also coordinating the communication of that information, that is locking, using a clock, using the rhythms to establish the clock that can establish timing for shared communication. These are basic principles. Sometimes it's referred to as sort of dynamic or functional connectivity, and that is you have anatomical connectivity, two brain areas that are actually connected by axons, but then you have functional connectivity in which two areas have their timing such that output of one area actually arrives at another area in a form that can actually be processed. So you can use the timing to coordinate and regulate computation that involves communication. But then there's a third function, and that's one that I'm gonna focus on here, and that is that sort of in addition to well, it's kind of captured in these others. In addition to having, sort of the presence of an oscillation, you can characterize it by the frequency, by the power. You can also describe the phase, and that is this timing information. The relative phase of an oscillation can be used to lock different events. You can use phase locking, again, to do coordination between areas. You kind of lock the timing of these oscillations to coordinate the communication, but also phase within an area can be used to structure the timing of individual events. So much like the description of the sort of the discrete timing, right? Time step 1, you integrate. Time step 2, you transform. Time step 3, you transmit. Well, you can think of that timing as also being distributed across phase. Phase 1, you compute. Phase 2, or phase 1 you integrate, phase 2 you compute, phase 3 you transmit. So you can use phase channels as a way of both separating the timing and organizing the sequencing of computations. And so what I'm going to show you is that that's precisely what the hippocampus seems to do. It does all three of these things that I mentioned. And that is the organizing of local computations, the coordination of communication of that computation to other brain areas, and the structuring of computation using phase, not just phase locking, but sort of differential phase-dependent computation, different functions at different phases. Now, one of the reasons for bringing this up, I think, is that we're sort of thinking about synthetic systems, not just biological computation, but also thinking about synthetic or machine learning, machine computation. I like to point out that one of the areas that I feel is sort of currently underrepresented in areas of AI and machine learning is in the area of sort of dynamic computation and temporal coding in general. So thinking about how you use time rather than, time is simply something that has to accommodate computation. You use time to perform computation. Using time to perform computation is very common in analog systems. Analog computation makes use of time. Time-varying signals perform computation. The simplest computation, I always like to point out, is something that when I was a graduate student at Caltech and Carver Mead, who was really sort of the founder of this kind of the analog VLSI movement, he liked to point out that analog systems are capable of computing, for instance, transistors will compute derivatives. They compute derivatives naturally. Of course, derivatives are a part of any computation. Any computation requires derivatives. Analog systems, they compute them instantaneously, naturally, for free. You get derivatives for free. If you do this digitally, of course, you have to do approximations. You do some sort of discrete iterative approximation to a derivative. So it can be computationally intensive. So if you were to use a system, exploit the natural abilities of analog computation to give you certain computations. When I say free, it's not necessarily free, but at very low energy cost, right? At the minimal expenditure of energy to perform a computation, you gain a significant advantage. Now, there's just the basic advantage that's often pointed to, brain consumes about as much energy as a light bulb, and all of this sort of thing. So there's sort of there's the energy efficiency argument, but I think that kind of misses another aspect of that constraint, the energy constraint, and that is when you are constrained to use very little energy and you need to exploit analog computation, you may find solutions that maximally leverage that, and that is you find novel solutions to what you otherwise might think of as conventional problems. So the brain has basically been forced to come up with solutions that leverage these principles of analog computation that give you the necessary efficiency. So again, I just sort of pointed out, this is the basic method. You get these spikes, you get spiking activity. Well, what can you do with the spiking activity? Well, again, pointing back to the early 1970s when John O'Keefe discovered the property of spatial firing or place cells in the hippocampus. And the approach they'd used was very simple, and that is you put an animal in a natural environment. In this case, a natural environment is described as an open field. It's just a little box. In this case, it's a box. This is an apparatus that I used when I was doing my first recordings of distributed multiple single unit extracellular recordings of hippocampal activity. Put an animal in a little box like this, record the activity, in this case of like 80 neurons simultaneously over about 10 minutes. In this case, using a color code to indicate the average firing rate. So you just like accumulate, so forget about like the spike timing stuff. This was early, in the early days, we're just recording spiking activity. And here the primary measure was what's the firing rate? That's just, how many spikes were actually produced, and in this case, at a given location. So thinking about coding in terms of the number of spikes, so-called rate coding, this is the representation of the rate coding property of the hippocampus. And what you see here, for instance, each box or each panel represents the activity of a single cell. So there are about 80 cells here. Again, the spikes accumulated over about 10 minutes. And what you see here, this is each panel is a cell, and it's the top-down view of the box showing the location in the box where that particular cell fired. So for instance, this cell fired sort of in the upper right-hand corner. This cell fired in the center of the box. This cell didn't fire at all. These cells didn't really fire at all. They're blue everywhere. This cell seemed to fire all across the box. So each of these they're both different cells, but also kind of different classes of cells. So for instance, those that show very kind of spatially constrained spatial receptive fields like this, these are the principal cells of the hippocampus. The pyramidal cells receive input and serve as the primary output. The cells that seem to fire all over the place that you find in a lower number, those are the inhibitory interneurons, inhibitory cells that form primarily local connections within the excitatory network. And then these cells that are not firing, these are the blue panels. You say, well, how do you, how do you put a panel there if the cell didn't fire? How do you know that it's a cell? So these are cells that could be active. If I took this animal out, I put it in a different box, this cell would express a place field. In other words, it's a pyramidal cell. It's just not responsive in this environment. And the way you can detect these cells is, well, you could exhaustively put them in, repeatedly in different environments and ultimately find the receptive fields, or you could simply allow the animal to stop running and rest. During periods of rest, quiet wakefulness, or during sleep, the population of cells that could be available seems to become available. And as you see firing across cells during periods of quiet wakefulness and sleep that may not have been active during active behavior. And so you can think of it as kind of a latent population. There's this latent population that's back there. It's not active, but you can see it when the animal is not actively engaged in exploring the environment. Now, another property, so that's the average firing rate as a function of location. This was the property that John O'Keefe described, place fields, place cells. He was given the Nobel Prize for that discovery. Another property that O'Keefe also was described, which came not from just allowing an animal to explore in these open fields, but constraining them to run on these, what are referred to as linear tracks. These are narrow corridors. This is a top-down view of two of these so-called linear tracks. This one's a C-shaped track, about 2 meters long, a couple of meters long. And what you see here, now instead of showing you the average firing rate, you're actually seeing the individual spikes. So each dot here is an individual dot. It's just kind of accumulated over multiple trials. The animal runs back and forth for about 10 minutes. And what you see here is the emergence of what looks like the spatial specificity that I showed you in the previous panel. Yellow cell tends to fire here. Red cell tends to fire over here. The green cell fires over here. And then I put the animal on a different track, and what you see is the property that I referred to earlier, and that is these latent cells, the cells that are silent on this track are active on this one, the light blue and the purple cell. So you can get different populations in different tracks. You have the cells that are here that sort of fire in different locations of the track. But the one thing that you kind of miss here, and this is the property that emerges when animals run on these tracks, is that this yellow cell doesn't just distribute spikes here when the animal's in this part of the track. It only it produces these spikes when the animal's on this part of the track running in this direction. Runs through this direction, you get spikes. You get this kind of pop, 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 bunch of spikes. Peak firing rate for these hippocampal cells is about 10 spikes per second. And we'll see this kind of 20 spikes per second is related to the dominant rhythm that is expressed during running, the so-called theta rhythm, which is in about the 8 to 12 hertz range. So you'll have this rhythm, and as I mentioned, the rhythm can be used to coordinate the timing. The spikes will phase lock to that rhythm. And so when you're getting this kind of 10 hertz you're getting this 10 hertz firing rate, what it really means is I have a cell that's spiking every cycle of this 10 hertz oscillation. Sometimes you can get higher rates, not 10, but maybe 20 hertz. What you get there, or maybe somewhere in between, what you get there is instead of firing one spike every cycle, maybe you fire 2 spikes every cycle. So again, there are a number of features of firing that are missed, even with this, where you're seeing the individual spikes. The first of which is this directionality. Now, the directionality is particularly interesting because here, if you think about this, and you kind of think about the animal running on this track. Okay, there's the yellow cell, so running from here over to here along this path that goes from yellow, it'll activate the red cell, and then it'll activate the green cell. So you're going to get a sequence of firing, yellow, red, green. Now you can say, oh, wait a minute, Matt. What about that blue cell there? There's that blue cell. It's right at the same spot as the red cell. Well, I can tell just by looking at this that the blue cell isn't going to fire on that trajectory. And that is, it's directional, but it's directional in the opposite direction. The red cell will fire at this location in this direction, and the blue cell will fire in this direction. And you say, Matt, how do you know that? Well, okay, A, I made the slide, and I presented this a lot of times, but B, if I gave you a little insight into properties of place cell fire, you would be able to do the same, you would be able to sort of draw exactly the same inference. And that is that they're both directional and they have opposite direction movement. And the way I can tell is if you actually look at these place fields, they're slightly asymmetric. Now there's a kind of a tail. So you notice the red cell, it's got this little tail. As the animal comes in here, you notice there's a few spikes here, then it can grow stronger and stronger, and then you have this very dense area, and then it kind of drops off. In other words, it's skewed. The place field is skewed, and it's skewed opposite the direction of motion. This cell will fire, the red cell fires in this direction. The blue cell sort of has the opposite profile. So there's a lot of spikes over here, and then it's got a dense area here, and then it falls off kind of over here. So there's a kind of an asymmetry opposite the direction of motion. That's how I can tell which way. And so you could, you could maybe tell the same thing about the other cells. So for instance, this yellow cell, I'm not even trying to use the cursor, it's too hard. You say, which way does the yellow cell, what's the directionality of the yellow cell? Well, it could be actually, if you kind of look over there, it actually may be over in this direction. If I have to look at the tail, I sort of described it in this direction, because that's the way it's most convenient. But actually, if you look at the tail, it may be skewed in this direction. It's the skew that actually determines, you could say, that correlates with the directional bias. And we'll see where that skew actually becomes relevant in thinking about the temporal coding that can occur when you have cells that are directional. So when you have cells that are directional, what that means is at this location, if I tell you, oh, red cell's firing, you can tell me not only where the animal is, oh, it's over here, you can tell me what's going to come next, which way is it going. So if I tell you the red cell's firing, you say, oh, okay, red cell, that means green cell is going to fire next. If I tell you blue cell fired, you can say, oh, the animal's going this way, yellow cell is going to be firing next. So now you have not only spatial information, not only directional information, but you can use this to do predictive, you have this predictive information. You know where you're going to be in the next second, a few hundred milliseconds. So now you have real trajectory information. And so the directionality, if properly exploited, can give you this kind of sequential trajectory representation. And so we'll see that's exactly what happens. This directionality translates into unique trajectory or sequence representations. And I'm just going to show you this. This is the raw data. And this is just kind of point out that, the thing that I remember first recording in the hippocampus when I was actually when I was a postdoc. I moved in the lab of Bruce McDaughton and developed these approaches for doing the multiple electric recordings, my first recordings. And I didn't know anything about the hippocampus, didn't know anything about place fields. This was in the, the 1980s. And, oh, question, go ahead. Great question. And that's how quickly, the question is, so how quickly does for instance, the directionality, do the fields form and the directionality, the sequence dependence form? So we actually looked at that. Again, sort of the proxy of that would be the skewing or the asymmetry of the fields. As we'll see, other properties that arise from that would be the sequential coding, and that is how quickly does the hippocampus both recognize and encode the sequence of cell firing, as we'll see in these sort of phase-locked rhythms. And the answer is very quickly. Run trial seems to be enough. The first pass through there seems to be sufficient. And that's also consistent with another phenomenon that I'll describe to you, and that's not the encoding of sequential information, but the retrieval or the replay of that sequential information during these so-called quiet wakeful states. So the hippocampus really has these two modes: active exploration, where it's taking information, and then the quiet state, you can think of it as kind of a quiet reactivation state, where instead of taking information in, it's evaluating and expressing information. It's retrieving information. And in that retrieval state, how long does it take before the hippocampus can retrieve a spatial sequence that has gone through? And the answer to that is one trial. One pass through there, and you can see the sequential coding of spatial sequences, and when it stops at this location, you can see the retrieval of those encoded sequences. So it's like one-trial learning, which, that was the thing that the hippocampus was always argued to be critical for, one-shot learning. What is it about episodic memory that distinguishes it from other forms of memory? Well, the argument was, well, episodic memory, sort of by definition, only occurs once. You get one shot, right? That's the idea. You experience something, you don't get repeated, it's not like classical conditioning where you get, you see a sort of a cue and a reward, and you're able to, with repeated sampling, establish the contingency relationship. You get to experience everything in your life once, and you have to form memories of it. And so the idea was that's what the hippocampus does, fast, one-shot learning. There was another question. Yeah. Great question, great question. So, so I think that, the question is, what is really the independence or orthogonality of these fields? And that is that if you had, let's say, you had a geometry where you had like overlapping paths, you had kind of common points, intersection, bifurcation, how do those get represented? And you could say, in this case, directionality is enough to actually separate out all of the behavior in this case. If you were to, sort of analyze what's the real dimensionality of behavior in this task, okay, it's kind of effectively two-dimensional, 1.5, depending on what you would take the directionality, as. But suppose you add in, like you had like a Y maze, for instance, where now it's not just the direction, but let's say at the bifurcation point, you could be pointing in this direction, but maybe you go left or you go right. And so now there's sort of an additional dimension, let's say, where you have multiple intersections. What you find is that this property, the directionality, now expands to include those additional dimensions. And it expands to include them, and so that you get, rather than thinking about spatial dependency or directional dependency, you're really thinking about it, and this is the way you're described as trajectory dependency. And that is that the place spatial firing is dependent on where you are, where you were, and where you're going to be. In other words, you can think about sort of defining the dimensionality as being a temporal dimensionality. And the temporal dimensionality, if you just sort of map, what are all the configurations of past, present, and future state, that kind of defines, that's the dimensionality of the hippocampal representation. That's kind of an interesting project to think, well, okay, but how do you know? You sort of described it as kind of a three-point temporal representation, right? As, past, present, and future. But, what's the real dimensionality? Is it 3 points? Is it 5 points? You could analyze that. You could think, okay, let's sort of put it in the most, in the sort of the most complex, sort of time state, behavioral apparatus, and that would be the open field. You put in the open field where at any point, right, I could be here, but there are essentially an infinite number of future trajectories that I could follow, basically kind of biased by the, body kinematics, right? I can't like instantly teleport to any point. But you can ask, what's the complexity of the hippocampal representations in an environment like a 2D environment where you have kind of infinite trajectories? And so what you find is that the representation is, as I'll show you, really constrained by the temporal dependence of this kind of past, present, and future state within one of these theta oscillations. So there's a temporal rhythm. The rhythm sort of defines, this is the window over which we're going to represent stuff. I can organize information by phase so that you have like the sequence or a trajectory, and the trajectory within this theta cycle, that's the representation. That's the dimensionality, and the content of the representation is captured by that, what goes on in a single theta cycle. That's the idea. So it's like whatever you can do, and then just thinking about, well, what does that translate into an environment? Well, you kind of map it. 10 hertz, animal running at about like meter per second, clipping along pretty good. How far can you actually go in one theta cycle? And it's a couple of body lengths basically. That's sort of it. It's like where can I, you can think of your kind of memory as being constrained by what am I going to be doing in the next, the next second or two, couple of seconds. Sort of that's the time window. I sort of describe it. Nobody watches MTV anymore, but it's the MTV model of memory, where the idea of the MTV is like little short, brief, quickly edited snippets. That's the idea. You have that experience is sort of formed, broken up into small chunks, each chunk lasting no more than a couple of seconds, and that's constrained by the biophysics. Yeah, there certainly are changes. So there certainly are changes with experience. But this kind of comes back to this question, how long does it take to form these fields? It's basically one pass. One pass, and you can tell where the cells are going to fire. What changes over time is the consistency of firing, and that is, if I look here, from here to here, it's going to take it maybe a second. So that's like, a second to walk along that. Again, at about a meter a second, it's going to take about a second. That's going to be like 10 theta cycles. So you get like 10 cycles, and you can ask, like with experience, what happens is you get more consistent firing on individual theta cycles. That's what you get. And so when you actually get to the peak of firing, like here, the first pass, you run through here, maybe you get some firing, you get a couple of spikes. They'll describe that as the firing rate is low. What it really is the firing, the consistency of firing at each of the 10 theta cycles over this field is relatively low. And when you do it 10 times, now you start getting more consistent firing at each theta cycle, which means every single pass now starts looking more and more. Now, you can describe this as a change in firing rate. And very often, this change in the firing, either as a function of environment or context or as a function of time, is described as remapping. So remapping used in this context is appropriate, and that is same input, different output. So for me, that's sort of the definition of remapping. I have the same input, but I get different output. So the changes in firing rate as a function of time describes what's referred to as rate remapping. That is, you can have changes in firing rate that occurs, because of experience. When you put an animal here, for instance, and you get a different pattern of firing, the literature uses the same terminology, which is not, I don't think it's really accurate, but still it gets used. This remapping, this is referred to as the population is remapping, but it's referred to as global remapping. So it's different, the cells fire in different locations or different cells fire. So change in firing rate in a given environment, rate remapping, change in the distribution, the spatial location, or the sampling of cells that fire in an environment is called global remapping. Now, I would argue that if you have different input and different output, I'm not sure where the remapping, this is just mapping, right? Global remapping is just mapping. Rate remapping is, in fact, some change in the transformation. I have the same input. Why do I have a different output? Well, I have remapped it. I've changed the mapping algorithm so I get a different firing rate. And as we'll see, if you look at single cells, you see this as an apparent change in the consistency of firing with respect to, let's say, the theta rhythm. When you look at a population, you see something that's very different, and I think that's where you see sort of the real effect of experience, and it's really the consistency of firing across cells, and that is the covariance of firing across a population. So just some numerology. In the rodent hippocampus, you have about 100,000 cells. On average, at any given point, you have about 1% to 5% of those cells that are actually firing, which means you have typically maybe, let's say, a couple of thousand cells. At any given time, a couple of thousand cells firing. So you can think of the hippocampal representation is this, it's a vector, about a 1,000-dimensional vector that's actually telling you, it's giving you a code, unique code across 1,000 cells. And so the question is, well, how do you read that code? Of course, the code across, 1,000 dimensions, well, they all have to be, all the elements have to be sampled, equivalently. If some of the cells are not firing, then it's not really that useful. So to get a really consistent 1,000-dimensional code, all 1,000 of those cells have to be firing. They have to be firing basically at the same time. And so what you find is when you look at a single theta cycle, when I say, oh yeah, with experience, this cell tends to fire more consistently across theta cycles. What I'm really saying is, with experience, this cell and the other 1,000 cells that are firing with it tend to be firing more consistently, which means if you're reading out this 1,000-dimensional code, you'll actually see, oh, look, these are the 1,000 cells that are active at the location. I'm really confident that's, that's the location. If this cell only fires, kind of sparsely, it doesn't fire every time, then the consistency of that code goes down and you have a sort of a less reliable output. And so this actually makes two important points, as we'll see later. One, that the firing rate, the increasing firing rate on a population level will translate potentially into increased covariance, which means you can use it to reflect confidence. Let's say it can be like a recognition code, familiarity code. Just, yes, I am really sure that this is, this location. I've seen this location before. And so that's what you find, that in fact, with experience, that familiarity with an environment can be read out, can be decoded by simply looking at population covariance. I don't even need to know what it's coding. I simply have to ask, well, if I see very consistent firing at this location, every time it goes in here across 1,000 cells, I can be pretty sure the animal's been here before. I don't know, I can tell that it's familiar. On the other hand, if I get variable firing, low covariance, that's an indication of relative novelty. Now that's important because that can be used. You can think, look, if I'm downstream, I'm the prefrontal cortex, and I don't know what I don't know what the actual code, the spatial code of the hippocampus is. All I want to know is this familiar or is this novel? I want like a novelty or familiarity code. You can think of this just universally, not just the hippocampus, but anywhere. You can use that property, the covariance, the fact that you have firing that's going to be constrained by these dynamic oscillations, that you take a cycle, one cycle, and you say, okay, each of the cells in that cycle, they're all going to be like independently sampling whatever the inputs are. And if they all agree, yes, on the output, then you're going to get a consistent, you're going to get a consistent output. And you can say that that consistency is going to come from, let's say, having prior experience that strengthened the synaptic inputs, that covariance across a population is going to be a reflection of like synaptic modification or specific learning on the inputs that were associated with that. So you can kind of infer learning by looking at covariance. And covariance is going to be reflected at the single cell level, this kind of consistency of firing with respect to these oscillations. So I've almost forgotten what the question was, but I think I tried to answer it. Right, exactly. So you could say, that's the thing about like the one shot, right? Episodic one shot, it's always novel. You get exactly one, you get one sample. So the idea of familiarity, relative familiarity with respect to like an episodic memory, doesn't really make a lot of sense, right? What does that mean? So, what I've described, relative familiarity, is like another property of experience. And so what we'll see is that the hippocampus is capable of actually performing two simultaneous forms of coding. One, a sparse sequential code, which really captures the order but doesn't give you any indication of like this relative familiarity. Temporal coding. And then there's this rate coding. It's really a sort of regime, it's a phase regime, where now the rate code can be used. It doesn't give you any kind of sequential information. So in other words, it can't capture the episodic nature of experience, the one-shot nature of experience, but it can capture this relative familiarity, and that's the repeated sampling. And so you can think of that as, that's like the recognition component. It's like when I'm here, I can say, I've been here before. Or I come up to here, I'm trying to, what building do I go in? I come up to this building and I say, I could either remember, oh yeah, I remember last year I came and I went up to this building and this was where I gave the lecture. Or I can say, this just feels like the right spot because this is familiar, right? The familiarity component, I don't have any specific memory of coming in, but I can say, I've been to this building before. Why? It just feels like I've been there. That would be the rate code, the covariance code that would have only formed with repeated sampling, the fact that I'd been here like 5 times before. Whereas the episodic code, one shot, no covariance, now it's the sequence that I'm actually retrieving. And we'll see that those two coding properties are actually segregated. They're done simultaneously. They're segregated in phase within the theta oscillation, which is sort of a clever way of kind of solving those two coding problems, familiarity, one-shot episodic coding. So this just shows I kind of showed you wait a minute here. Yeah, this is just the raw data. I didn't actually get to show the raw data, but so I show you this both to kind of illustrate that this property, the spatial firing, it's just something that comes out in the raw data. And as the little story I was telling, the first time I was recording the hippocampus, the thing that strikes you when you just listen to this activity is just, so how clear this correlate is. You just hear it in the raw data. It's not like you have to, run statistics on it to see the spatial coding. And then the other is that you can hear the dynamics. So you can hear the dynamic modulation, which is expressed when the animal is running, but then you also get to see what happens when the animal stops. So there are really two modes to the hippocampal activity. The active behavioral mode, which is indicated by this rhythmic modulation that you'll be able to hear, and then there's this quiet wakeful mode, the stationary, when the animal stops. In the active mode, you get play cells. The quiet wakeful mode, the animal's sitting, you get something else. The rhythm goes away, you get different kind of code, but you can see it all in the raw data. And so I'll just play it for you so you can kind of see, and I'll sort of narrate through it. So you kind of hear the activity. Yeah, the volume might be a little low. I don't know if I can turn it up. All right. So here you see like play cells being expressed. So this was like the question first time through. It takes one pass. You can kind of see the spatial test. I'm not saying this is the first time the animal ran through this, but you can see it in a single pass. Now here the animal's going to stop. Now if you wait, there's a little pause. And then you get a little burst of activity. It's very characteristic of the transition from the active exploratory mode, in which you get this theta rhythmic modulation, which if you listen, it sounds kind of like this. You listen really carefully. Well, he's going to stop here. A little burst. If you listen, I listen to it a lot, so I can hear it very clearly. But if you listen to it, you can actually hear the modulation of the population with this rhythm. That's the theta rhythmic modulation. So theta when you're running, this kind of bursting activity when the animal is stopping. And so this is just showing exactly the same data, but now instead of just showing the raw data, we're going to use this sort of Bayesian decoding. So you're just using the sort of the conditional probability of firing. Place field is just the conditional probability of firing given location. But then you can also invert that using Bayes' rule to compute what's the probability of the location given the firing. That's all. So you're just using Bayes' rule to ask what's the likely location. So at every time point, you ask which cells are firing, and then given the previous sampling of the place fields, say given that pattern of firing, what's the likelihood that the animal's at any given location on the track? So you just figure out the conditional probability of being at any location on the track every time point. And then you represent that probability as a triangle, size of a triangle. And so this is the continuous Bayesian decoding of population spiking activity in the hippocampus. And what you see is, same data. So when the animal's moving, you get this blue triangle, which tracks the animal's position. So it's a place code. I can decode the animal's position, in this case, every 100 milliseconds. Pretty accurately. I mean, the probability of being anywhere else is essentially zero, no triangles. But interesting, when the animal stops, triangles go away. I can't tell where the animal is anymore. But during those bursts, you see that the triangle jumps around. What that means is that you're decoding, and when you decode the activity within those bursts at high temporal resolution, you see that the code that's expressed corresponds to remote locations. So the code during active behavior, animal present location, code during stationary behavior are remote locations. And then as we zoom in closer, we'll see that that code has a finer kind of temporal resolution than that. It's not just present location when the animal's moving, it's past, present, and future, and that is that there's kind of a local trajectory, and that the stationary reactivation also has trajectory information. It's remote, not just remote locations, but trajectories that pass through remote locations. Okay, so, and that when you take the animal off altogether, when you're sleeping, you get the same kind of reactivation. That is, the hippocampus can retrieve. When it does retrieve information during sleep, it takes the form of these, it takes the form of these sequences. The one I illustrate here just has sort of an interesting property that we will touch on, and that is, in this case, the sequence runs backward in time. So these sequences can run both forward and reverse in time. Yeah, question? Yeah. So that's just very simple. So you just sort of compute the posterior distributions by sampling the place fields. So they run through it. They already run through it, and then you can either go back retrospectively and then decode based on the previous sample, or you can use, field sampled in one half to predict activity in, another half. But it's in the same environment. It's just sampling the place fields in the environment and then using those fields to decode the position. That's what I'm saying. I mean, you're kind of double dipping. I mean, you can kind of split it. You could sample in one session, decode in another session. You can do that. I mean, it just sort of depends on the stationarity. It's not, yeah. I mean, the application during the behavior, sort of decoding the position using the positional fields, we can say, well, okay, it's kind of double dipping. The real use of that was to decode like the activity during the non-behavioral states, non-active, like the quiet wakeful states, the reactivation states, and the sleep states. That's the real value of it. But you can use it to decode the actual positions, just to illustrate that it does correspond to the animal's position. Now you'll see that you can use the same kind of sort of average firing rate posteriors that you generate by just measuring place fields, and then you can use that to decode at finer temporal resolution, which we'll see here, within the theta oscillation, and there you can, then it actually gives you value, because it's not double dipping. It's using the average firing rate, encoding model to predict the sort of within theta cycle decoding. So that gives you what we'll see here are what are referred to as theta sequences. So yeah, ultimately you want to use that, use the encoding model derived from like firing rate over behavior to decode other states. It could either be within theta or it could be like in the quiet wakeful reactivation. That's the real value of it. So this is just to illustrate, and we'll break very quickly. This is sort of illustrate these two behavioral states, two dynamic states within the hippocampus that correspond to the two distinct behavioral states. On the left, you see this some work from Yuri Busaki, where he's recording the local field potential, and that's sort of the average synaptic, the dipole generated by synaptic input in the hippocampus. And over here, when the animal's moving, what you see is this is the theta oscillation. So this says that the synaptic inputs and then sort of the corresponding spiking output is locked to this 10 hertz rhythm. When the animal stops, is stationary, within about a half a second or so, you go from this oscillation, theta oscillation, to these kind of spikes, large irregular spikes. This is an older nomenclature. This was referred to as large irregular activity. You can see by sort of looking at that why they called it that, large irregular activity. When you zoom in more closely, it's not shown in this figure, what you see is that these kind of big extracellular spikes actually have two components. One is the big spike part, which Busaki termed a sharp wave. It's kind of a wave. It's longer in duration, about 100 milliseconds long. There's a big deflection. It's sharp. And then on top of this, there's a high frequency oscillation, about a 200 hertz oscillation known as a ripple. And so these events were referred to as sharp wave ripple events because of those two components. And so you have sharp wave ripples, which are associated with immobility, sitting quietly, and also sleep. And then the theta oscillation, which is associated with active locomotion, and interestingly, another stage of sleep, REM sleep, rapid eye movement sleep, which is also referred to as paradoxical sleep. The reason it's called paradoxical sleep is that that's when you dream, and that's where it kind of, brain activity looks a lot like you're actually awake. It's paradoxical in that you're asleep, but your brain looks like it's awake. So not surprisingly, it's in that paradoxical sleep state where you see the theta rhythm. So you can think of theta rhythm as being associated in some way with kind of active exploration of sampling of the environment. This quiet wakeful or sharp wave ripple state as what I refer to as an offline state, and where it's no longer, you're no longer trying to process or encode information coming in from the outside, but you're trying to reevaluate information that you've already collected in some way, reactivating prior state information. And so this is just, we'll get back to this, we'll break quickly. So this kind of gets into now the temporal coding story. So there was the place fields, there's the directional or kind of potentially sequential encoding dependency of hippocampal firing, but then there was also this temporal coding, the dynamic modulation by this these rhythms. In particular, the theta rhythm, which you can see here, is about a second of activity, in this case, in 2 brain areas. In red, you see the hippocampus, and in black, you see simultaneously recorded the prefrontal cortex. And this is just to illustrate 2 things. One, you see the lower trace, the local field potential, which again sort of reflects the activity of these synaptically driven dipoles. This is sort of the input. So the input is modulating, is sort of varying this 10 hertz rhythm. And then you see on top of that, spiking activity, the output. The output you can see is sort of phase locked to the input, not surprisingly. And that is spiking phase locked here in the hippocampus in red. In this case, at about the peak of the theta oscillation. Now, interestingly, if you look up here at the top, these are spikes generated in the prefrontal cortex. Completely separate area. Yeah, they're synaptically connected. There are connections between them, but functionally they're quite distinct. But you see the spikes in the prefrontal cortex. This is just a single cell in the prefrontal cortex. Spikes in this prefrontal cortical cell are locked to the theta oscillation in the hippocampus. You say, why the heck would the prefrontal cortex care about the timing of activity in the hippocampus? Well, you would say, well, maybe if the prefrontal cortex was trying to communicate to the hippocampus, maybe it's listening to the hippocampus, and maybe you sort of infer that based on the relative timing. Well, the hippocampus fires first, and so this idea of phase locking and coherence of activity in these 2 structures, suggesting maybe there's some sort of interaction, we'll kind of visit later on to see that, yes, indeed, there are these transient periods during which the prefrontal cortex and the hippocampus will phase lock to the theta oscillation, suggesting that, that phase locking is actually predictive of effective decision making, so that it seems to actually be used. Interestingly here, you can see that you have phase locking of the prefrontal cortex with the hippocampus, even in the apparent absence of the theta oscillation in the prefrontal cortex itself. And so we can kind of revisit what that actually means. But again, it suggests that when you see these oscillations, the presence of the oscillation suggests that there's some sort of coordination of the synaptic inputs. Lots of common input coming, at the same time, which is why you can pick it up in the local field potential. The phase locking is what really tells you. So you need the spiking to tell you, oh, the prefrontal cortex may actually be listening to the hippocampus, because you can't get this just by chance. There's got to be some common signal. Either that's common input, in which case you would see coherence in the oscillations themselves. Same input, right? Same input, same oscillations. But in this case, you don't necessarily see the same input. It suggests that these prefrontal cells, they're not all being driven at the same time. They're not all being driven by the theta rhythm. It's just some of them might be listening to hippocampal cells that are modulated by the theta rhythm. And so that's what you see in the prefrontal cortex. A lot of heterogeneity, different cells doing different things. Some of them might be listening to the hippocampus. The way you can tell whether they're listening is by looking at the spike, either the sort of the spike correlation or spike coherence, or the spike to field correlations, spiking in one area, phase locked to the fields in another area.